Protein structure 8. Okay, so we're going to talk more specifically about protein shape now. This figure from your textbook, figure 4-11, shows a huge variety of different protein shapes. And this is just showing a few snapshots of different protein shapes. There's certainly many more out there. And basically that's one of the main points here, is that there's a huge variety of shapes and sizes This variety is due to the fact that there are 20 different amino acids which are found in cells, and due to the chemical properties that vary across those amino acids. So one question for you is, so which type of model are we looking at for all the proteins that are over there in figure 4-11? And the answer is it's a space-filling model. So you can see kind of the surface of the protein, but don't see a whole lot of details. There's two general categories of protein shapes that are shown here. One of those is globular, which sounds like a made-up word. So globular proteins, these have a compact, often rounded shape. And if you take a look over here, do any of them jump out at you as globular? So for me, let's see. Cytochrome C, that's looking pretty globular. Chymotrypsin, also looking pretty blob-like. Insulin, and deoxyribonuclease. By the way, this DNA here, that is not a protein, but just shown to make it a little more interesting. So basically, those are all enzymes. That's very typical for globular proteins to be enzymes. The other major category of protein structure that's shown over in that figure are fibrous proteins. And just like it sounds, it would be some sort of a fiber. They usually span long distances and are pretty skinny molecules. So if you look over there, do any of the proteins jump out at you as being potentially fibrous? Hopefully collagen jumps out at you. That is a fibrous protein. And as we might expect, these long, skinny structures also often have structural roles rather than catalytic roles. So kind of building materials. So fibrous proteins, these often have structural roles. And maybe you recall hearing about collagen in another biology course, or you might have heard of it in terms of bodily enhancements. So collagen is sometimes injected, say in lips, to make lips look larger. And so collagen also has a similar role in terms of structure for cells. It's part of the extracellular matrix, however. So that's one of the roles for fibrous proteins is extracellular matrix, one of the examples. And a second example is the cytoskeleton. We are going to see 3 different examples of fibrous proteins shortly, one of which is filaments. Second is to form sheets. And third, to form spheres. And in particular, the filaments are the very long structures. Okay, let's talk a little bit about these proteins that will assemble into potentially structural roles. And so this is an assembly of proteins. This is when we have several identical protein subunits that then will bind to each other and form a new structure. So basically here we see this example, these little green half kind of crescent things. Each of them has a little binding site, this little divot here. And so if there's just one binding site, then they basically can kind of hold on to each other, and then this makes a dimer. So that's one example about how we can start to make some assemblies or assemblages of proteins. Another example over here in panel B is that there might be two binding sites. This little divot and this little kind of peg thing pointing out. It kind of looks like a pop bead if you ever had those or built something with them. And basically now that there's two binding sites, and so we're going to use that same building block over and over again. It looks like maybe about 20 times. So basically that means that the next unit can bind with its little plug to that little divot and then just make a huge long chain here. And so this might make a helix structure potentially if this has some curvature to it. And a third possibility is that if the binding sites are just a little kind of more constricted on this one side rather than perfectly across from each other by 180 degrees, then they might make a smaller structure like this ring right here. And so one reason we're talking about this is as we especially talk about the cytoskeleton later in the course, we're going to hear more about these binding sites and how particular units can be assembled over and over again into a repeated pattern to make some larger structure. All right, so right now we'll talk a little more briefly about how we can take those subunits and build these three different structures: a filament, a hollow tube, or a spherical shell. So first off, let's talk a little bit about the filament. An example of the filament is actin. So actin, this is one of the main parts of the cytoskeleton. It has very long fibers. Actin is made up of these protein subunits repeated over and over again, and they assist in constriction of the cell membrane. And so they function kind of like a drawstring. So a drawstring, let's say it's on a pouch, it can just tighten up just a little bit around the edges and then kind of close off a particular portion of the cell. And so actin can do some kind of shrinking and moving like that, but basically of the membrane ruffles. And again, we'll hear more about that later. For now, it's just an example about actin. Actin kind of sounds like action, by the way, so that might help you think about that constriction. Here's a second shape that can form from subunits, and that is a hollow tube. And an example of that is tubulin as a subunit. We can have many tubulin subunits. Again, these are proteins, and then they can assemble into a hollow tube, and then we call that whole long tube a microtubule. So basically, in that example, each of these little dots would be a tubulin protein, and then this whole thing is a microtubule. So we're zoomed in really close from if you've seen microtubules under a microscope or in other drawings before. The microtubules are also part of the cytoskeleton, and these are found in a different location, and they have a slightly different function in the cell. They instead hang out more in the cytoplasm, and they basically can provide three-dimensional support for the cell. And a third example that's shown here is a spherical shell, and this is when we have many individual subunits that can glom together in a different repeated fashion. And one example about how spherical shells are used is in viruses. Some viruses will have a protein coat around them, and that can be a spherical structure.